second ring, and by team 4 in one ring outside. And one nice feature of this channel is further that within the hexamer, always 2 subunits have C-termini oriented in an antiparallel manner, as you see it nicely here. And this antiparallel manner is on the one hand required for STIM1 binding to the ORA channel. It mediates a signal to the pore, so that the pore activates. And this antiparallel orientation is maintained by 2 hydrophobic residues, so 2 leucines or 1 leucine or isoleucine, independent of it's the Drosophila structure or the human structure. So I would like to explain to you or give you insight how we got to know these 2 proteins and how they function. And on the one hand, we use the patch clamp technique. On the other hand, we use the thread technique. So the patch clamp technique we have heard several times during this meeting here. We are using here the whole cell mode. It's rather well known. And for that, the pipette is tightly attached to the cell, and you can measure the current across all ion channels in the plasma membrane. As we are measuring here store-operated calcium ion channels, we need some substance in the pipette which leads to this passive store which leads to store depletion. We are using here EGTA. It's a well-known calcium chelator, which lowers the calcium concentration in the cytosol and leads then to passive store depletion. And outside the cell, we are using the calcium ion as we are measuring calcium ion channels to measure the calcium entering the cell. In the beginning of the experiment, we just attach the pipette closely to the cell, that we have access from the inner of the pipette to the inner of the cell, and let EGTA diffuse into the cell. To measure the calcium ion currents, we are applying voltage ramps from minus 90 to plus, or here minus 95 to plus 75 millivolt. And in the beginning, all channels will be in the resting states because the stores are still full. So you will just see one line close to 0. But then stores will get depleted after some time, and then you will see some activation of the current, as it starts then here. So you see some activation of the current. From each trace, you take 1 point at the same time, so 1 current at the same time, and you see this nice time course where currents activate already after 30 to 40 seconds to a maximum after 100 seconds. So this is a typical time course of a store-operated calcium ion channel, and this curve you see here is a typical IV curve of those calcium ion channels. It just shows current into the cell, almost nothing outside the cell, so it's nicely inward rectifying and shows that it is highly calcium selective. On the next slide you see if you just co-express, so we are using here the standard expression system hex cells. If you just express STIM in hex cells, you get no current. If you just express ORI in those cells, you also get no current. Only when you express both STIM1 and ORI1, you get nice store-operated activation. So these 2 proteins are essential, but also sufficient to lead to this typical store-operated calcium entry. Another nice feature of those calcium ion channels is the fast calcium-dependent inactivation. It's a negative feedback mechanism within the cell, which guarantees that not too much calcium can go into the cell. You will see it here. So it can be measured by applying not now a voltage ramp, but a voltage step here from 0 millivolt, in this case, to minus 90 millivolt. And then you will see that currents activate very fast, and then they start to inactivate. And you see a typical inactivation trace for the STIM ORI currents with the step from 0 to minus 90 millivolt, very high currents. It's here normalized to 1. And then it inactivates very fast to also within 50 to 100 milliseconds. And after these 50 to 100 milliseconds, it's typical for those STIM ORI currents that it shows some reactivation. The reason for that is currently not really known. It's not visible in endogenous crack currents, but it's a typical feature of the STIM1 ORI1 currents. So the other technique we used is the thread technique. With this technique, you can detect very nice protein-protein interactions. We are using here fluorophores, such as GFP, but in this case, CFP and YFP, coming from the jellyfish, Aquaria victoria. And the two fluorophores need to have emission excitation spectra which are rather overlapping. And you can detect a FRET signal when the two fluorophores come very close to each other in the range of 10 nanometer. And when you attach those two fluorophores to two proteins, as it's done here, and they are far apart from each other, you can only excite the donor fluorophore, here the CFP, and detect the light of the emitted light of the CFP. But when the two proteins come close to each other, the fluorophores can come close to each other, and the radiation-less energy transfer can occur from the excited donor fluorophore to the acceptor fluorophore, in this case the YFP, and you can detect via excitation of the CFP light from the YFP. And in this case, you talk about the FRET signal, and you can measure protein-protein interactions or very close coupling of two proteins in the cell. And within the cell, a pixel-per-pixel readout allows you time-dependent measurements of FRET changes. And we use those two techniques, the patch clamp technique and the FRET technique, to understand better how STIM and ORA are working and how the signaling machinery works. So in the beginning, in the resting state in the cell, calcium is localized in the ER. Calcium is bound to this calcium sensor, the EF hand, in the STIM protein. And the STIM proteins are organized as dimers. But when calcium is depleted, STIM proteins start to oligomerize, so they form higher order aggregates, and this can already be nicely shown with the FRET technique. For that we used CFP and YFP and labeled the STIM proteins with these two fluorophores. And in the next slide, you see a co-expression of CFP STIM and YFP STIM in the cells. And the first row is here the resting state. This is the overlay in the FRET image. And in starting from 2, we added tapsigagen, a circuit pump inhibitor, which leads to calcium store depletion from the ER. And you get altered localization. You see it nicely in 4 and 5 of the STIM proteins, so they are localized more in clusters, and an enhancement in FRET. This correlates also nicely here with this diagram. You see this enhancement, and it also correlates with a calcium indicator in the ER, which shows upon depletion of calcium, reduction of calcium, a reduction in FRET. So STIM proteins form stable homomers after store depletion with tapsigagen. So what happens in the next step? The STIM aggregates now couple to the ORA protein. This can be also nicely visualized again with the FRET technique. For that, we labeled the two proteins, now both at the C-terminus with YFP and CFP. And here you see hex cells co-expressing now the CFP ORA, YFP STIM, the overlay, and the FRET in the resting state. When we apply again now tapsigagen to deplete the stores, we get again this altered localization of the STIM protein. It's more in puncta compared to here. The ORA protein also moves somehow in puncta, as you see it here and here, for example, and an enhancement in FRET. And also in the magnified image, you see from 1 to image 4, the strong enhancement in FRET. So STIM to ORA coupling is temporarily as well as spatially correlated with an increase in FRET. And what happens next? Calcium can go into the cell, but prior to this, we first wanted to know if some domain of the STIM protein is already sufficient to couple to ORA and to activate those channels. And for that, we cut the STIM protein and we just expressed the STIM1 C-terminus without the transmembrane domain, without the EF hand. And you see here, when it's just expressed in hex cells without ORA, there's a homogeneous distribution. But when it's co-expressed with ORA, you have nice localization in the plasma membrane, suggesting that it already goes to ORA independent of store depletion, and also some FRET. And this correlates nicely with current measurements. So when STIM1 C-terminus is alone in hex cells, you get no current. But when STIM1 C-terminus together with ORA1 is co-expressed, you get already currents independent of store depletion, so just from the beginning of whole cell break-in. So the cytosolic fragment is sufficient for aura channel activation, and we later found that even smaller fragments are sufficient for aura activation. But I don't want to go in detail here. So next, calcium, when the stim-aura complex is formed, calcium can enter the cell, and this requires a conformational change of the aura channel. This could also be monitored by fluorescence microscopy, but for that, rather single molecule fluorescence microscopy would be required. But initially, we and also other groups made studies with site-directed mutagenesis. And you see here, again, the 4 TM domains of the aura protein. And one might expect, if the aura channel opens, the TM1 domains just delayed and opened the pore. Doing site-directed mutagenesis, we found that when mutations are done in this TM1 domain, we get constitutively active currents which destabilize the closed state. But further, via site-directed mutagenesis, we found out that not only in TM1, but also TM2, 3, and 4, we get mutants which also become constitutively active. So there are several positions throughout all TM domains which lead to the activation of the channel or which are involved in keeping it in the closed state. And there is one mutant, P245L. It's localized in TM4, and it's associated with a disease, the Stone-Morgan syndrome, where people have problems with blood cells. It shows, in contrast to this diagram here, already activation from the beginning, so independent of stores. If you co-express STIM there, you get further activation, but basically it's already active in the beginning. It's the same for all the other mutants, so we suppose that the aura channel undergoes a global conformational change upon activation, and not just a change within the TM1 domain, the pore lining domain. So summarizing till here, what is required in the STIM-aura activation cascade? Initially, it's ER store depletion, which leads to the activation of the STIM protein. Next, STIM-aura coupling. And finally, a conformational change in the aura pore, or in the aura complex. But I would like to continue now with the role of STIM in aura activation, and further also with the coupling domains in aura 1, which are essential for binding of STIM1. So first of all, the role of STIM1. You have seen in the previous slide here that STIM1 can further activate the constitutively active mutant. So basically it's required for store-operated activation, but it has several further roles. There are typical crack channel characteristics, and they are listed here. So for the wild-type STIM1 aura activation, you see here typical store-operated activation in a calcium-containing solution. But when you switch from this calcium-containing solution to a sodium-containing solution without any divalent ions, then you get strong enhancement in currents. Another typical feature of the crack channels is the strong inward rectification, so no current goes out, and it's highly selective and has a reversal potential in the range of 50 millivolt. And further, also the fast calcium-dependent inactivation, as I have shown before, also this typical feature with this 50 to 100 milliseconds of inactivation is typical for crack channel hallmarks. If you have a look now on the constitutively active mutant we had before, when it's co-expressed with STIM, you have similar behavior. You have those enhancement in currents in the sodium-containing solution. You have a reversal potential here in the range of 50 millivolt and nice inward rectification. And you also get this typical fast inactivation trace, as it's shown here. But when it's not co-expressed with STIM, it looks different. You have not this increase anymore. You have a reduced reversal potential and an altered shape in this IV curve. And you have no inactivation anymore, you have just reactivation. So STIM1 is required to produce all those typical crack hallmarks, or hallmarks of those store-operated ion channels. And how is STIM doing that? For that we investigated now the coupling domains within aura 1 required for binding to STIM1. So we continued again with the wild-type aura protein, and we once deleted the C-terminus, once we mutated the C-terminus. The C-terminus contains a binding domain, the so-called coil-coil domain, which is known for many proteins to form protein-protein interactions. And you see here in both cases, for the deletion mutant and the point mutant, loss of function. So when it's co-expressed with STIM, there is no activation anymore. And also in the FRET images, so this is the resting and the store-depleted state, you get no enhancements in FRET. So STIM1 or one C-terminus is one essential binding domain for STIM1, and if STIM1 is not able to bind, then also those crack hallmarks are not anymore there. That can be nicely visualized with this constitutively active mutant. But in the next step, we also wanted to know if other cytosolic domains within ORA are required for STIM binding. So we continued with the N-terminus, and you see here a deletion of the N-terminus, again leads to loss of function. But in this case, you get enhancement in FRET. And a further look on the co-localization of the ORA protein together with a STIM1 fragment. It's a STIM1 C-terminal fragment, which is already able to bind to ORA independent of store depletion. You can monitor co-localization of this fragment with ORA1. You see here, this is outside the cell. The peak here is the membrane, and this is the cytosol. And for ORA1 wild type, you see very high intensity just around the membrane, showing that STIM1 C-terminus is interacting or is nicely co-localized with the ORA protein. For delta C-terminus, for this deletion mutant, there is no co-localization, homogeneous distribution throughout the cytosol. But for this deletion mutant, it's somewhere in between. That means STIM1 is still able to bind, likely through the C-terminus, but it's somehow affected by the N-terminus. So the next step, we wanted to know in as far the N-terminus is now really required for coupling and activation of ORA. It is of note that among the ORA channels, there are 3 isoforms, and within the N-terminus, they have a region close to the TM1 domain, which is fully conserved. It's the ETON region, extended transmembrane ORA membrane region, and it is a helical region extending into the cytosol coming from the TM1. And we did sequential deletions of this region, and we find that deletion till 75 still leads to preserved store-appropriated activation, even if it is already somehow reduced. But deletion to 76 or 78, so till here, leads to complete loss of function. So residues from here of this ETON region are required for store-appropriated activation. And again, in respect to STIM1 binding, it's the same like for the full deletion. So deletion till 78 and 76 are somewhere here in between for this co-localization of STIM protein with the N-truncation mutant. So also those residues are required for STIM1 binding. So in the next step, we again had a look on those typical crack hallmarks. Specifically here now, this enhancement in current from a calcium-containing solution to a sodium-containing solution. And you see here, for all the functional deletion mutants, which is shown here in the calcium-containing solution, one of those, the 74 deletion mutant, which is active, but in the presence of sodium, it shows a reduction in currents. So for maintaining the typical crack channel hallmarks, it's even required, even more of this N-terminal region is required for crack channel function and STIM1 binding. In a further step, we wanted to know how does it look for other ORA isoforms. I mentioned before that this region in here is fully conserved. So one would expect that it happens the same for ORA2 and ORA3, but this was not the case. As you see it here, for ORA1 deletion mutants, 76 and 78, you get loss of function. But analog deletions in the isoform ORA3 show preserved function or even higher function. And we asked what is the reason therefore, even if this region is almost fully conserved or fully conserved. So we performed or constructed several chimeras, and we found out that the region between team 2 and team 3, a flexible region in here, this loop 2, is responsible for this difference. And you see here, The deletion mutant shows loss of function, but when you add there the ORA3 loop 2, you get again enhancement and function. The same is for the ORA3 mutant. When you add there the ORA1 loop 2, you get loss of function. And what is the reason therefore? So at the level of, we examined this now at the level of ORA, just at the level of ORA with this constitutively active mutant, and you see there the P245L mutant is constitutively active. When it loses function upon deletion, but when you add there the ORA3 loop 2, you get again function. So somehow the loop 2 is, the ORA1 loop 2 is inhibitory in ORA1 deletion function. And what is the reason therefore? We performed several MD simulations and we find that two residues here, or at least two residues, come only in the ORA1 truncation mutant in very close distance in contrast to the wild-type ORA1 or the ORA3, both full-length or deletion mutant. And the reason for that is that this flexible region between the team 2 and the team 3 is longer than for ORA3. For ORA1, it's longer than for ORA3, leading to inhibitory interactions with team 1. What's the effect now for wild-type ORA1? The main thing is that there are two residues, 78 and 166, oppositely charged, which can come in close proximity. And the ORA channel itself, or also its binding to STIM1, somehow adjusts this communication, because when you do a cysteine cross-linking of those two residues, you get reduction in current and a release of those cysteine residues, again enhancement in currents. So already at the level of ORA, this communication affects somehow ORA activation, and a right communication between N-terminus and loop 2 is required to guarantee normal activation. So the N-terminus is finally required for store-operated activation, for maintaining typical crack channel hallmarks, and for a communication within the ORA channel. But finally, we also found that the ORA N-terminus is also involved in cholesterol binding. There is a well-known cholesterol binding motif, and when we do both chemical depletion of cholesterol via cholesterol oxidase, but also mutations of well-known residues which are involved in cholesterol binding, we get always enhancement in currents. Also here enhancement in currents, and also here enhancement in currents. And these mutants are already insensitive to cholesterol depletion, because when you add there in addition the cholesterol oxidase, you get no further enhancements. And interaction studies here with top fluorocholesterol and ORA1 wild type showed that there is nice thread between top fluorocholesterol and ORA1 wild type in contrast to those mutants. So finally, the N-terminus also contributes to binding to cholesterol and regulating STIM-ORA activation via cholesterol. So in the end, I would like to conclude. The STIM proteins exist as dimers in the YAR membrane in the resting state. When the stores are depleted, the STIM proteins form higher order oligomers. They couple to the ORA channel and activate calcium into the cell. And the main binding site is now ORA1 C-terminus. And the ORA1 N-terminus has several roles, as I mentioned before, coupling to STIM1, store-operated activation, controlling crack hallmarks, controlling a communication with other domains within the ORA channel, and it's further involved in cholesterol-mediated regulation. Finally, I would like to thank the groups which are all involved, the group with the head Christoph Romanin, and our collaboration groups of Klaus Koschner, Mitsu Ikura, and Rudi Ettrich, and finally also for your attention.
A lot of work, a lot of data, and nice conclusions. Are there any questions?
Hi. Do you have any idea about the role of cholesterol, or does it induce a conformational change in ORA, or what do you think?
We assume that it induces a conformational change. We have seen several mutants within ORA1 which induce constitutive active currents, and we assume that this cholesterol somehow affects the arrangement of the TM domains, which also leads to somehow activation or somehow to an active channel. So cholesterol depletion or the loss of cholesterol binding leads to the dilation of the pore somehow.
Okay. Do you think that also the properties that cholesterol confers onto the membrane, like membrane fluidity or so, can affect the ori?
It's possible. It's known for many other ion channels. I mean, in this case, based on further data, we know that, or we think that there is really a direct binding of cholesterol with ORA1.
Okay.
Visibly no more questions. Then we can move to the next talk. Model membrane.
Okay, thank you. I'm Pavel Hruboučák, and I came from Joint Institute of Nuclear Research from Dubna, near Moscow. And I would like to introduce you our study, which was devoted to location of general anesthetics in model membranes. Alkanes, as well as aliphatic alcohols, are well known for their anesthetic properties, but the origin of this anesthetic effect is still unknown. It is supposed that it could be induced by the interactions between the constituents within the membrane. On the other hand, what is well known is that the change of membrane structure has significant influence on their functional properties. So we set an objective to investigate the influence of decaying molecules on the change of membrane structure, hence the change of specific functions of biologic membranes. And here is the illustration of a biologic membrane. And as you all certainly know, it is very complicated and complex structure. It contains a lot of features which are interacting with each other, and it's the mixture of phospholipids of different kinds. And due to its very high complexity of real membranes, we decided to study the properties of membranes on model membranes. So we created bilayers which were as simple as possible. They were composed just from one kind of phospholipid. We chose DOPC, and in these model membranes we tried to introduce decaying molecules as the representatives of general anesthetics. According to previous studies, there is a correlation, between the amount of decaying in the bilayer and between its distribution and even its orientation. And according to these previous studies, two theories were proposed. First is claiming that molecules of decaying are concentrated in the center of the bilayer, and they are oriented with the perpendicular to the lipid chains. And another theory says that molecules of decaying are concentrated below the lipid heads, and the molecules are oriented parallel to the lipid chains. As you can see, the orientation and the concentration of decaying molecules has an impact on the thickness of the membrane, for example. So in order to distinguish which of these theories is correct, we decided to label the molecules of decaying and to find their actual position within the membrane, within the model membranes. Let me tell you a few words about the samples preparation. At first, we created we diluted DOPC molecules and molecules of decaying in organic solvent in order to obtain the homogeneous mixture. And such solution we spread it on the wafer of silicon, and employing a rock and roll method, we were in glow box. we were slowly evaporating the solvent, and in the end of this process, we obtained a regular structure of thousands of parallel bilayers. This is what you can see on the picture. And before the measurements, all the samples were hydrated by water. And in total, we obtained 4 couples of samples with labeled and unlabeled molecules of decane of different scattering contrast. Once we had the samples in the form of multilayers, we were able to perform diffraction experiments. But instead of X-rays, we utilized neutrons. And the reason for that was that atoms of deuterium scatter neutrons significantly different than the atoms of hydrogen. And this is also the principle of deuterium labeling of decane molecules. So from diffraction experiments were performed in France, in Grenoble. We obtained the diffraction patterns, as you can see on the picture. And from diffraction patterns, we obtained curves like this. So from data analysis of diffraction patterns, we were able to say that our samples were of high quality because we were able to recognize the diffraction of 5th order. And from diffraction patterns, we were able to reconstruct the bilayer profile. This is because there is a relation between the distributions of measured scattered intensity and the parameter which is called scattering length density of unit cell. And scattering length density is characteristic for each particular structure. And this is good for us because according to this, we can recognize these structures within the bilayer. So as you can see, the scattering length density of heads is different than the scattering length density of end of tails, for example, and so on for each particular structure. Here I would like to demonstrate how we obtained the actual positions of decane molecules in membrane. So we took the profile of the membrane, which was corresponding to the sample containing unlabeled decane. This is the blue one. And we simply subtracted this profile from the profile corresponding to the sample containing labeled molecules of decane, the green one. And the red curve is the difference between those profiles, and it corresponds to the distribution of the label within the model membrane. And I hope this picture is quite convincing for you, that I can claim that the molecules of decane are concentrated mostly in the center of the bilayer. So let me conclude that according to our analysis, according to our study, for decane to DOPC concentration 1:1, the molecules of decanes are concentrated in the center of the bilayer. And this conclusion is corroborated not only by the analysis I have already shown you, but also by the simulations of molecular dynamics, which we performed recently, and they ended up with the same results. And in the near future, we would like to investigate the different concentrations of decane and its influence on molecules' location within the membrane. Thank you for your attention.
So it was very convincing, but in the case of a real biological membrane, there is no guarantee that the decane will be somehow in the center because there are proteins and other objects. But labeling the decane by deuterium provides an opportunity to put into real biological membranes also and to do the same measurement, not?
Oh, sorry, I didn't understand. Correct.
So if you would prepare membrane preparations from real biological membranes, then you could study there also the location of the decane in the membrane. Okay, it is more difficult because you cannot have easily so regular layers, but it is not impossible.
Yes, you are right, but if we want to investigate the changes in the structure, it's better for us to do it on model membranes because it's not confusing. But of course, you are right. We can track the position of decay also in real membranes.
Yeah. If I understood correctly, so you demonstrated that the concentration of decay in the middle of this bilayer is higher than at the edges, yes?
Yes,
That's what
you're
this
right. According
scattering,
to our analysis.
I would like to ask that whether you were also able to see that there was a difference between the width of the membrane.
Yeah, we also observed the change of thickness of the membrane. We have already performed the experiments with different concentration of decays. I have no data here, but I can tell you that there were the difference between the thickness of the membrane when we put another amount of decay.
Thank you again. On calcium membrane interaction.
So hello, my name is Adela Melstrova. As already said, I come from Prague, and I would like to tell you something about the influence of transmembrane proteins on the affinity of biological membranes to calcium ions. Calcium in cells has many roles. The main role should be signaling, and also it has a role in membrane fusions. And for this purpose, its concentrations in various compartments of membranes has to be really maintained. And for example, in endoplasmic reticulum, there are millimolar concentrations of calcium, whereas in cytosol, there are hundreds of nanomolars. But what is not really known, what is the concentration of calcium ions in membranes? So we focus on this topic, and it is really crucial to know the calcium concentration there because calcium signaling occurs really nearby the inner leaflet of the plasma membrane. And when there are these bursts during the signaling events, the concentration range of calcium can be really different. And the signaling events are always or are often in these caveolae or domains rich for cholesterol and sphingolipids, and also there are transmembrane proteins and peptides for sure. And the calcium binding influences the structure, function, and also oligomerization of membrane proteins. What is already known, we previously studied recently calcium binding to pure phospholipid membranes, and how does the membrane biophysics changes when the calcium ions actually bind there. And to go further in more biologically relevant models, we also investigate the cholesterol, sphingolipids, and in this particular case, the membrane peptides. So to summarize the recent study on pure phospholipid membranes, as the first really crude model for inner leaflet of plasma membrane, we had POPC membrane, and then we added 20 molar% of negatively charged POPS, and we identified the binding sites there, which are phosphates. In a case of PS, these are also carboxylic acid groups, but the calcium ions do not just go only to the phosphates, but they penetrate a little bit deeper, and they are bound also to the carbonyl. And they are like co-bound between these few groups and also between a few phospholipids surrounding the calcium ions. And the ions reduce the membrane hydration, so the membrane is dehydrated a little bit. The regions at phosphates at carbonyls levels are a little bit rigidified, and the internal interlipid distance at the lateral interlipid distance is also reduced. So the membrane is a little bit shrinked, I would say. And to investigate the peptide influence on the calcium affinity of the membrane affinity for calcium, we designed 2 artificial transmembrane peptides. Both of them have this leucine-alanine sequence in the middle, then 2 tryptophans, and the positively charged one, VALP-KK2+, has 2 lysines at each end, so it's 2 plus positively charged at each end, which is biologically relevant because there exists something so-called positive inside rule that the excess positive charge of the transmembrane proteins in plasma membranes is really in the inner leaflet of plasma membrane. And this positive charge, we expected that it might repel calcium ions and thus reduce the affinity of the membrane for the ions. And then we have a zwitterionic peptide with lysine and aspartic acid at each end, and this one should not attract or repel calcium ions. And we raised these questions: what is the influence of transmembrane alpha leaks on the calcium binding, and if there is if there is the impact of this positive charge of the peptide on the binding of calcium ions. We access these questions by the fluorescent methods, which is time-dependent fluorescent shift. And it works like this, that we have a fluorophore, we use Laudant probe, which is located at the carbonyl level of phospholipids. By a laser pulse, we excite the fluorophore to the excited state, and the molecules around, in this case, the phospholipid molecules around, they sense the changed electric dipole moment of the fluorophore, and they reorient themselves to be in more favorable positions for these changed electric dipole moments. And this reorientation takes some time, and there is a spectral shift, like the energetic drop in the energy of the excited state. We can measure these 2 parameters and directly get the local hydration and mobility or rigidity of the molecules around. These measurements were accompanied by zeta potential measurements, and this is important to say that zeta potential doesn't probe the surface charge, but rather the charge nearby the membrane at so-called slipping plane. So we sense also not only the rigidity and hydration inside of the membrane, but also the charge nearby the vesicles we use for our studies. And at this graph, you can see the results from the fluorescence experiments. On the y-axis, there is a relaxation time, and the higher the relaxation time is, the more rigid is the membrane. You can see that if you add the non-charged peptide to pure POPC membrane, the membrane is rigidified a little bit. But when you add the positively charged one, the rigidifying effect is higher, which is a little bit counterintuitive because the membrane itself is not charged, so why the positively charged peptide could have a bigger impact on it. But it does, and for PCPS membrane, the trend is the same. Also, the positively charged peptide has a bigger rigidifying effect on the membrane. To these models, we added calcium ions, and you can see that always the calcium ions, when we go from 0 to 200 millimolar, it has additional rigidifying effect. And it is important to note that this additional rigidifying effect of calcium in percentage is the same regardless the peptide content. So the calcium is not repelled by this positive charged, and it does not care if the peptide is present or not. The binding affinity of the membrane is always the same. For this, I also performed the zeta potential measurements, and the POPC membranes, they behave like expected. The non-charged peptide didn't change too much the charge nearby the surface. The positively charged one indeed increases the charge. However, the PCPS membrane didn't behave like this. Also, the non-charged peptide increased the zeta potential. For this, I performed also test experiments with a different negatively charged lipid. So I exchanged POPS with POPG, which is also negatively charged, and the other one behaved like this. So this behavior is POPS specific, and from this we hypothesized that it is associated with some POPS group reorientation, which could somehow shield the negative charge of PS. So when we add also the non-charged peptide, the PCPS membrane, the charge on it is changed. Then we added also the calcium ions there, and the results confirmed the observations from the fluorescent technique that the calcium binding binds to all the system regardless the peptide content or its charged. And with this, I would like to just conclude that we see the rigidifying effect of both peptides. The positively charged one has a always bigger impact, and the non-charged peptide increases zeta potential at PCPS membranes, which can be accompanied by some POPS head group reorientation. And for this purpose, I also am currently running MD simulations to really see the molecular interactions, to see the reorientation of the PS head group, if it is really there. And the membrane affinity for calcium ions is not affected by the peptide presence, and there is no suppressing effect of the positive charge of the peptide on calcium binding. And this is my group in Prague. Martin Hoff is our boss. My work was consulted mainly with Piotr Jurkiewicz here. And I would like to also thank the funding, especially Martina Rezlova Memorial Fellowship I was awarded this year, which provides me daycare for my 2 children. And I would like to thank you for your attention, and I'm ready for questions.
Thank you very much. Question? Yes, please. Alexander.
Do you know what is the capacity of phosphatidylserine for calcium? I mean, how many of these molecules?
Okay, so this is the work we already published in 2016. So for POPC, we see the capacity like 3 lipids per 1 calcium. So it's 1 calcium per 3 lipids in POPC. And for PCPS membrane, we observed 1 calcium per 2 lipids. However, these numbers are a little bit over-exaggerated because we have these numbers from MD simulations, and we used a model which we know now that the binding affinity for the ions is a little bit over-exaggerated there. So it can be something between 1 ion per 2 or 3, something between.
Well, if there are no further questions, then we can just move to
Wow, okay, it's not good. Does anybody have Mac, maybe, to help me with the screen, because it cuts the... Okay, so hello. I'm very sorry to be your last lecturer for today. It's only me standing in your way for lunch. So I will talk about structural basis for NLP toxicity. I come from Ljubljana, from National Institute of Chemistry. And I will just shortly introduce the biological problem of NLP proteins. It was very nicely explained in the morning in the plenary presentation of my mentor, Gregor Anderluch. Anyhow, plants are constantly confronted with a variety of abiotic and biotic stresses in nature. And that's why they have to defend against these stress factors. But unlike animals, they defend against unwanted stress factors on a cellular level. Not like animals, because animals have a systematic system immune response. So cells of plants are equipped with so-called PRR molecules. These are receptor molecules, and they recognize specific patterns on microbes or the proteins that those microbes produce. So after the recognition, this is a so-called pattern-triggered immunity. Gene expression comes, and the plant cells are defending against stress factors. So one type of proteins that plant recognize are NLP proteins. These proteins activate this complex, but they are secreted by several phytopathogenic microorganisms, and they are found in fungi, bacteria, and oomycetes. But they have dual roles, so they activate plant immune system, but they also cause leaf necrosis in dicot plants. And in 2009, three-dimensional structure was solved, and they found out that the structure was very similar to actinoporins. And actinoporins are cytolytic proteins that act by disrupting the cell membranes of target cells, of course, and upon specific binding to sphingomyelin. And because of this structural similarity, it was proposed that these two protein families share a common cytolytic pathway. So instead just of recognizing a small part of those proteins by PRR, so by these receptors, NLP proteins probably act directly with the membrane, and they disrupt the membrane. So our research was divided in 3 parts. So we had to identify the lipid receptor, then we performed interaction studies, and I will mostly talk about the structural analysis performed with X-ray crystallography. So as Gregor already mentioned in the plenary presentation, the target molecules for NLPs in the plant membranes are so-called glycosylinositol phosphoceramides, and they are constructed by ceramides with phosphate and the polar head group, which contains a variety of sugars. So we found out that NLP proteins bind to the whole molecule, to the whole GIPC, but we were very lucky to hear that NLP proteins also bind to this sugar residues at the end, so the terminal sugar residues, in millimolar affinity, but it was still good enough for us. So the NLP proteins bound to glucosamine and monosamine. Okay. And this is the result. So this is the crystal structure of NLP proteins. In gray is the apple form of NLP protein, which was the structure that was already determined in 2009. And we solved 2 complexes of NLP with glucosamine and NLP with monosamine. And you can see that the overall fold stays the same, but there are some small conformational changes in the structure. This is the electron density of the sugars. It is not, if anybody does X-ray crystallography, it is not really a perfect electron density in comparison to the rest of the protein, the electron density of the rest of the protein, but we could still say that this electron density belongs to the sugars because we could see that in asymmetric unit there are 4 polypeptide chains, and only the ones that had this additional electron density had structural changes that govern binding. And you can see this, one of the changes is this conformation of the loop L3 in both cases. And another conformational change is the coordination. So of this magnesium ion, as Gregor said in the morning, the very important part of this protein structure is this coordination site. And in apple form, this magnesium ion is coordinated by this set of amino acid residues, while in complex structure, this magnesium ion shift towards the interior of the protein. So this magnesium ion gets coordinated with a different set of amino acid residues. And you can see this histidine 101 is directly involved in the coordination, but when the binding of the sugar occurs, this histidine is free. And this histidine then later interacts with the sugar that came in the structure, okay? So besides this histidine, there is also aspartate 158. So we performed point mutation of this binding site, and we also observed this change in conformation of loop L3, and we saw here that there is a conveniently placed tryptophan residues, and the toxins that interact with the membranes usually use tryptophan residues for anchoring. So when we change this tryptophan residue to alanine, we saw that if this is a wild type necrosis, if we change this tryptophan residue to alanine, we saw a decrease in toxic activity. So that means that the protein was not able to anchor in the membrane. We also performed point mutation of this aspartate residue here, and when we changed this aspartate to alanine, we saw no decrease in toxic activity. That means that the sugar could still come in because alanine has a smaller side chain. But when we changed the aspartate to bigger residues like glutamate, lysine, phenylalanine, and others, we saw a decrease in toxic activity because all of a sudden side chain was in the place of the sugar, and probably protein could not bind the sugar head group of GIPCs. We prepared recombinantly all these proteins, and sometimes even the smallest change in the protein is crucial for protein stability and protein secondary structure. So we checked that these point mutations are really different from the wild type only because of point mutations and not thermal stability and secondary structure, and they were all the same. So wild type and all the mutants have the same secondary structure and they have the same thermal stability. And this is just another presentation of structural changes. So this is the apple form NLP protein. This is the cavity to which sugar binds, but after sugar binds to this cavity, you can see structural changes. You can see that protein opens up. And because of this change, the protein is able to embrace the GIPC head group. And this is a model we propose. So again, apo form NLP protein and the bound form of NLP. So you can see the structural change and this tryptophan residue that helps anchoring the NLP protein. But what we can see here is that this sugar head group is just big enough for this protein to embrace. But for instance, dicot plants have mostly this type of GIPCs in their membranes, but monocots that are insensitive to NLP proteins have longer chains or they're even branched. So because of this different type of GIPCs present in the monocot plants, the NLP proteins probably do not bind to them and do not cause necrosis. And just short conclusions. So we were talking about natural killers of plant cells, and we provided in this study the structural basis for NLP plant membrane interaction. We also explained the NLP dicot plant selectivity, and because these NLPs often attack economically important crops, this is very suitable for phytopharmaceutical development in the future. And with that, I would just like to thank all of you for listening, but mostly, of course, my mentor Gregor Anderluch, Marietka, Katja, Vesna, Tina, the whole department, and the German group we collaborated with. And if you have any questions, please let me know.
Laci, it's huge.
This was a very nice explanation, geometric explanation, so I was wondering if it would be possible to modify these proteins to increase the size of the cavity so that also monocut, with the mutant monocut plants would be affected.
Yes, but maybe you could, but I'm not sure why would you do that. Because monocuts are insensitive. You don't want diseases on plants.
For a further proof of what you're saying.
Okay, yeah. Well, it was like Gregor was saying, there are some monocuts that have this type of GIPCs, and if you treat those kind of plants with NLP proteins, they are infected, and they have leaf necrosis. But I don't know if you could change it. You should change the cavity to make it bigger, but I'm not sure. I mean, what's the purpose?
Well, if there are no further questions, then there is nothing more to let. It was the last lecture. So thank you for the lecturer, and thank you for the audience. And we have to thank the organizer.
Enjoy the lunch, and before you leave home, of course, safely. I would like all of you to thank you to make this event. Without you, there is no event, of course. And RBC continues, so I'm pleased to invite Hungarian friends, Professor Zimani. Yes, please, make an announcement for the next one.
Just a few words. We are really grateful for János and his team, and I can assure you that the tradition is not broken, will be not broken. So in 2020, the next RBC conference will be held in Hungary somewhere. We are already thinking about it where, but let's keep it as a secret. There is no decision. What will be difficult is to match the standards set by our Slovenian friends, but we'll try to do our best. We'll see. But this was really an excellent conference, so it's not an easy job. So please watch for announcements sometime next year. And on behalf of the board of the Hungarian Biophysical Society, I would like to invite all of you in 2020 in Hungary. Not all of you, but everybody in your societies who are not here now. Watch your spam folder. Make sure that our announcements don't land there. And see you in 2020. Thank you.
Thanks, and we are, of course, looking forward to Hungarian RBC meeting. And with that, I would like to close this RBC and thank you all for being here.